Hi, welcome to our channel of IGNOU Audiobooks, Indira Gandhi National Open University, School of Agriculture, SOA, Certificate in Beekeeping, CIB, OAPI 01 to Management of Honeybee Colonies, Block-1 Bee Health Management, Unit-1 Beeenemies and Their Management, 1.0 Aims and Objectives. The aims of this unit are to times familiarize yourselves with important insect pests associated with honeybees and times acquaint you with the various management practices to contain these enemies. 1.1 Introduction. Like our crops and animals, honeybees are also infested by various pests and diseases. In this unit, we shall describe the characteristics of important insect pests attacking the honeybees. The diseases of the honeybee shall be discussed in the next unit. Aristotle, 384 to 322 BC, was the first person who described certain disorders in the honeybees. Until 1800 AD, any outbreak of pests and diseases in honeybees tended to be confined to a limited area only. But in recent times, the quick mode of transport and movements of bees and bee products from one location to another across countries and continents have aggravated the pest problems. There are reports about the incidences of large number of insect pests infesting honeybees worldwide, including India. In India, apiculture is a very young science. Research on bees was initiated in early 1940s. Within such a short period, several enemies of honeybees have been studied from India. Bee enemies include insects, mites, spiders, birds, woodpeckers, bee-eaters, honey buzzards, etc., and mammals, bears, skunks, badgers, baboons, etc. Wax, moths and parasitic mites are endemic in our country. We shall take up one by one the major pest of honeybee. 1.2 Common insect pests of honeybees. Let us examine the features of the important insect pests associated with the honeybees. 1.2.1 Greater wax moth, Galleria melonella. The greater wax moth, Galleria melonella, is a serious pest of honeybees. The adult wax moth is grayish-brown and has a wingspan of around 3 centimeters. The adult female moth enters into the hive and lays eggs in the cracks and crevices of the hive. It also lays the eggs directly on the comb. The wax moth larvae feed on the wax comb and occasionally the pollen. The larvae move towards the anterior of the combs by continuous feeding. The larvae move near the midrib of the combs in silken tunnels woven out by the larvae from their secretions. While feeding, they also excrete minute elongated black striated pellets. Such feedings weaken the comb. The heavy infestation reduces the comb into web, mass containing comb pieces, silken galleries and excreta. The exact nature of damage to the beehive can be seen in the fig. 1.1 The wax moth does not attack on the bees as such. Its full development to adult requires access to the used older combs or cell cappings which contain proteins essential for the larval development. Wax moth infestation is higher in hot and humid conditions and is more serious in weaker honeybee colonies. This pest is more serious on Asiatic bee species than on European honeybees. The surplus wax combs stored in super chambers is also infested by the wax moth. Control. A strong honeybee colony generally needs no treatment against the wax moth. The bees kill and clean out the moth larvae and their webs, keeping the colonies stronger, removing extra surplus combs from honeybee colonies, keeping cracks and crevices plugged, and frequent cleaning of floorboard help in checking the wax moth infestation. No chemical treatment against wax moth is recommended. In the live colony in stored combs, wax moth can be controlled by spraying them with spores of Agave variety of Bt Bacillus thuringiensis. It is an effective biological control agent and has an excellent safety record. Storing the surplus combs in cold storage followed by their exposure to the sun is also helpful in checking the moth infestation chemically. The moth can be controlled with para-dichlorobenzene, PDB, moth crystals. Exposure of the stored combs with burning sulfur fumigation or with aluminium phosphide fumigation is also helpful in the control of this pest in the stored empty combs. After chemical application, the combs must be welled out for several days before their use. The use of naphthalene balls, moth balls, is discouraged because it accumulates in the wax, kill bees and contaminates honey. 1.2 to be predatory wasps, Vespa orientalis, Vespa auraria, Vespa mandarinia, Vespa tropica, Vespa basalis. The bee predatory wasp is a common enemy of honeybee in all the Asian countries. Among the most frequently reported species of wasp is the social wasps of the genus Vespa. Colonies of both Apis cerna and Apis mellifera are frequently attacked by these wasps. Wasps' invasion in A. cerna colonies generally causes the bees to abscond, and similar behavior is also reported from weaker colonies of A. mellifera. These predatory wasps catch the bees from the hive entrance and inside the hives. It makes the colonies weaker. Predation by Vespa spp on commercial apiaries is generally a seasonal problem. These pests are also active in hot and humid environment. Learning activity 1. Identify some common insect pests of honeybee in your locality. Make their inventory. 2. Identify some common non-insect pests of honeybee in your locality. Make their inventory. 3. Also identify the common methods to control the pest menace. Hint: you may interact with a progressive beekeeper of your locality. The wasp attack on apiaries reaches on peak during monsoon and autumn, July-October. Apiaries situated near the foothills and tropical forests suffer more acutely than those in the plains. Initially, a hunting phase is observed during which a few wasps capture and kill slow-flying bees one at a time near the entrance of a weak hive. Later, a slaughtering phase sets in. In this phase, some 20 to 30 wasps attack a weak colony en masse, using their strong jaws to maul the bees. The dying and dead bees are dropped to the ground. Finally, when this phase has continued long enough and have lost most of its defender workers, the wasps enter in the hive and eat away the honey and brood nest. Control. The foraging range of Vespa is comparatively in the larger area around their nests. Killing the gravid females during the spring season reduces the wasp population in the coming seasons. Bait trapping, trapping at the hive entrance, and using protective screens are helpful in minimizing their damage. Locating the nests of wasps by following flight passes of individual wasps back to their nests and then destroying the nests may though be time-consuming but is very helpful. Where labor costs are not prohibitive, capturing and killing individual wasps foraging in the vicinity of the apiaries is very useful in controlling the problem. The wasps can be killed by using fly, flapper or broom in front of the beehives, particularly during noontime when wasps are the most active. This approach has proved to be quite effective, largely because the period of most intense attack is only 2 to 3 months. Use of wasp bait in the apiary is also useful in minimizing the damage. Installing wasp screens in front of the hives also reduces the damage by keeping the wasps away from hive gate. In case of heavy attack, relocating the apiary is advised. 1.2.3 Black ants. Ants are very common insect. They attack on a wide range of stuffs. Various species of black ants viz. Camponotus compressus, Monomorium indium and M. destructor intrude in the beehives and take away the honey and pollen. They are more troublesome to weak colonies, but stronger hive usually succeeds in keeping them at bay. As black ants live in underground nests, their colonies can be destroyed by fumigation. Bee colonies can be saved from intrusion of ants by placing the hives on stands with their legs in water vessels. 1.2.4 Mites. Parasitic mites are the most serious enemy of honeybees. In tropical Asia, the success or failure of beekeeping with APIs mellifera depends largely on the mite control. Several major factors accentuate bee mite problems. For example, all the major species of parasitic honeybee mites are present in Asia, most being native to the continent. The complete eradication of the mites from an apiary is impossible because the feral nests of native bees infested by the parasites serve as reservoirs of mite. Moreover, some mites are able to survive or even thrive on more than single species of host bee. Several species of mites have been reported from A. mellifera and A. cherna colonies throughout Asia, though not all the species found within the hives or in association with the bees are serious. Many species of pollen-feeding mites are occasionally found in hives or attached to foragers. The mites are usually of two types depending upon their mode of action. If they attack on the outer body of the honey, they are referred as ectoparasitic mite, and if they ate affecting the internal organic of the bees, they are called as endoparasitic mites. Now let us examine the important features of the mites. 1.2.4.1 Ectoparasitic mites. 1.2.4.1.1 Varroa mite, Varrosis, Varrojacoxonial varroa destructor. This mite is a native ectoparasite of A. cherna throughout Asia and was first reported from Indonesia in 1904. Since the beginning of beekeeping with A. mellifera in the continent, it has been causing damage in both temperate and tropical Asia. The overall effect of varroa infestation weakens the honeybee colonies and thus decreased honey production. Occasionally, IGNOU mellifera and more frequently in A. cherna, heavy infestation may cause bee absconding. Today this parasite is found throughout the world except for Australia and New Zealand. In temperate Asia, most beekeepers agree that varroa damage is a constraint to the success of beekeeping with A. mellifera, while in tropical Asia, success is limited by the loss of A. cherna colonies through absconding, which is far less serious and frequent than damage to A. mellifera. Varroa jacobsoni multiplies faster on APIs cherna, whereas varroa destructor is more serious problem of APIs mellifera. Occasional removal of A. cherna, male brood combs, and keeping the hive in healthy condition are the ways of prevention of varrosis. IGNOU cherna colonies identification. Varroa destructor, before the year 2000 known as Varrojacoxoni, is quite large and can be seen with the unaided eye. The adult female is more wider than being longer with size of 1.6 by 1.1 millimeters WXL. The mite is shiny reddish-brown in color. The body is dorso-ventrally flattened, covered with short hairs. Setae. Adult females of V.de structure are found inside brood cells or walking rapidly and obliquely on combed surface. Individual mites are often seen clinging tightly to the body of adult bees, mostly on thorax or between the thorax and the abdomen. Adult males and the immature stages of both sexes, egg, larva, protonymph and deutonymph, are not seen outside the brood cells. All immature parasites live inside the brood cells. They can be observed when infested cells are opened and The brood is carefully removed. The immature mites are bright white and the adult females are brown, while male mites are smaller than females and are pear-shaped and creamish in color. Symptoms of infestation. Varroa causes injuries to honeybees by direct feeding. The adult bee is also affected if the infestation is severe. If more than one parasitic female mite infest the brood cell, the brood decays or adult deformations occur, including shortened abdomen or deformed wings. Generally, the bee brood die at pupal stage and can be seen as uncapped brood, commonly known as bald brood. Due to the mites passing through open brood cells, the larvae may die, which is seen as displaced and larvae attached erectly to the hive cell walls. Such larvae also have grayish spots on their heads. The displaced and cell wall attached larvae sometimes confused with the symptoms of European foul brood (EFB) disease. But these larvae do not lose their body segmentation or change their color so fast as observed in EFB disease. Infested adult bees often have a reduced fat body that hampers the functioning of their glands or increases their susceptibility to pesticides. The semen production of drones may be considerably reduced. Varroa mite is also a carrier of deformed wing virus, which causes shortened and twisting of wings. Unfavorable climatic conditions or insufficient stocks of pollen and nectar further enhance the process of disintegration. Without treatment, the colonies generally dwindle, and management errors may also cause the collapse of colonies. Mite detection. The most reliable method of detecting the presence of the mite is random opening of brood cells, particularly the drone brood cells. Remove the brood from the cells either with a fine forceps or by using Varroa fork and inspect it for the presence of the mites. You need to open at least 100 to 200 cells for imposition and assessment. To inspect adult bees, the bees are captured from the brood combs and placed in jars having chloroform or ether or alcohol-soaked cotton wool. The bees are intoxicated and the mites crawl on the glass wall. Returning foragers may also be captured at the hive entrance and held up against the sunlight. Any mites attached to the bees' abdomens may be seen. The mite can also be detected by installing Varroa board, which has 8 mesh size wire screen above and below. Place a sticky paper on the floorboard and observe after 2 to 3 days to detect the mites fallen and stuck on the sticky paper. The mite falling can be enhanced by using formic acid vapors at 5 RNL per day inside the colony, wherein 85% formic acid is used in injection vial with wick for 14 days. Control. Chemical control is the most popular among the beekeepers. It creates the risk of contamination of honey through accumulation of chemical residues in the hive and toxic effects to the bees. Among the commonly used mite control agents are organic acids, et hair oils, synthetic pyrethroids and amitraz. The application of chemicals can only be started after honey harvest. Let us examine the feature of some of the important chemical agents. Formic acid. Formic acid can kill the mites even in the sealed brood cells. An easier way to introduce formic acid is to use a sponge or any other absorbent. A solution of 3-5 Rn1 of 60% formic acid is applied onto the sponge tissue per comb, Langstroth size. The quantity must be reduced accordingly for smaller combs. The direct contact of bees to the formic acid should be avoided. The application can be repeated 3 to 4 times at the interval of at least 7 days. Oxalic acid. Contrary to formic acid, oxalic acid does not act via evaporation but through contact with the bees. 42 grams of crystal oxalic acid, dihydrate, is Thinned in 1 liter of sugar water, 1-1. Protective spectacles and acid-proof gloves should be used while handling the acid. 5 ml of the acid solution is trickled in between every 2 combs late in the evening. The treatment should be repeated 2 more times at an interval of 7 days. Lactic acid. Lactic acid is better tolerated by bees and does not cause problems in warmer climatic zones. The disadvantage of this acid is that every single comb must be taken out of the hive to spray the bees with the acid. The dosage applied per comb is 8 milliliters of 15% acid. This treatment can be repeated several times at intervals of 7 days. Thymol. Thymol can be applied as a commercially available ready-made preparation or in crystal form. 0.5 mg thymol is put into a gauze bag and deposited on the frames for some weeks, which will kill the mites emerging from the brood. Fluvalinate. Apiston strips. Fluvalinate is the active ingredient of commercially available apiston strips. It is a synthetic parathyroid applied as a contact miticide. It is highly effective and noted for its low toxicity to bees and ease of application. The strip can be suspended between brood frames for 2 weeks. 1.2.4.1.2 Brood mite. Tropy Elaps clary. Modern beekeeping with APIs. Mellifera in tropical and subtropical Asia frequently encounters infestation with Tropylelaps clary mite. The mite is a native parasite of the giant rock honeybee, A. dorsata, and is widely distributed throughout tropical Asia. Whenever A. mellifera is kept within the range of distribution of A. dorsata, mite infestation of the colonies cannot be avoided. Parasitism of A. mellifera colonies simultaneously by both the parasitic mites occurs rarely. The population of Tropylelaps often being greater than that of Varroa Tropylelaps might completely prevent multiplication of the Varroa mite. Identification of mites. Tropylelaps mite is much smaller than Varroa mite and can very easily be recognized with a naked eye. The adult female mite is light reddish-brown in color, with an oval-shaped body about 0.96 mm in length and 0.55 mm in width. The mite's entire bougeois is covered with short setae which can be seen only under microscope. The mites can be observed walking rapidly on the surface of the comb, generally walking on the brims of the cells. They are rarely found on adult bees. In all its immature stages, the mite lives within the brood cells of the bees, feeding on the brood's hemolymph. Fertilized adult females enter the cells before they are capped to lay their eggs. The stages of mite development are egg, then six-legged larva followed by protonymph, deutonymph and timely adult. Adult males of Tropylelaps do not feed because their chelicerae, the organs originally used for piercing the bee's integument, having been modified to transfer sperms. The life cycle of the mite is well synchronized with that of the host bee. Symptoms of attack. The damage caused to colonies by Tropylelaps infestation is similar to that caused by Varroa mite. Normally, the injuries inflicted on individual bees and bee brood are essentially the same. The abdomen of bees surviving mite attack is reduced in size and having a shorter lifespan than healthy bees. In heavily infested colonies, bees with deformed wings can be observed crawling about the vicinity of the hive entrance and on the comb surfaces, while pieces of dead bee brood evacuated from the hive by the house bees can be seen in front of the entrance. Hives severely infested by Tropylelaps shows an irregular pattern of sealed and unsealed brood. Since this symptom can be taken as a sign of a poor laying queen, the infestation by the mite must be verified. The best mean is to open sealed cells and inspect the pupae for the presence of the mite. If mites are present, Adult females can be seen walking rapidly out of the cells. To obtain a reasonably accurate estimate of the level of infestation, 100 to 200 brood cells should be opened and the brood pupae removed with forceps for close inspection control. API's mellifera colonies can catch the mite infestation from API's dorsata when latter bee rob the former bee colonies. You have to prevent robbing and drifting among the colonies in an apiary to check the spread of the mite, as the adult female mite can survive without bee brood up to 7 days. Only, its control can be achieved by creating brood-less condition in the colony for at least 7 days by caging the queen bee inside the colony. Chemical control, dusting of sulfur powder at 1 per comb is found effective. The sulfur should be dusted directly on the top bars of the combs. Chemotherapeutic measures described for the control of varroa are also effective for tropicilabs. Formic acid can be used successfully in its treatment as described previously. While handling formic acid, the user should wear acid-proof gloves and protective goggles. Application of amitraz is very effective when used either as a liquid spray on the surface of the brood comb and hive walls or as a hive fumigant in the same dosage. The treatment requires 3 to 4 applications at every 4-day intervals. All chemical treatments must be suspended at least 8 weeks before the onset of honey flow, and amitraz must not be used in spray form in the presence of open brood. Now let us examine the features of the endoparasitic mites. 1.2.4.2 Endoparasitic mite. 1.2.4.2.1 Tracheal mite. Acarapis woodii. This mite, Acarapis woodii, infests the tracheal system of adult bees of all the 3 castes. It was first reported in API's mellifera colonies in Europe in 1921. It was first reported in India from API's Cherna colonies in 1957 and from API's mellifera colonies in 1994. Presently, it is a very serious problem in North America. Identification: Acarapis woodii is a very small mite, 0.1 millimeters. It lives and breeds within the thoracic trachea of adult bees. The mite penetrates through Spirac-1s into the first tracheal pair of the thorax of 10-day-old honeybees. It lays eggs inside the trachea. Male offspring emerge after 12 days and females after 13 to 16 days. Symptoms of attack: the infested bees are unable to fly. They try to take off but fall down and keep crawling around the hive. Sometimes these bees can be seen climbing on the grass leaves. Close observation of these bees reveal K-shaped winged condition, i.e., forewing undhinned wing are not coupled, and due to this reason the beesre, unable to fly, infestation shortens the lifespan of the individual bees. The severe infestation of colonies causes them to lose strength and thus increases a colony's susceptibility to winter losses. The most reliable diagnostic method is the laboratory dissection. Samples of 20 or more crawling bees found near the hive and unable to fly are killed, their heads and legs are removed and their thoraces dissected for microscopic examination. The trachea are observed under the microscope. The mites are usually present at the end of the first pair of trachea in the thorax. Control: formic acid. Formic acid produces good results by applying it onto a cloth. 20 milliliters of 65% formic acid 4 times at an interval of 7 days. Use of 5 milliliters of 85% formic acid per day continuously for 3 weeks is also recommended. While applying formic acid, use acid-proof gloves and protective goggles. Treatment operation should be carried out during low humidity and when outside temperature does not exceed 30 degrees Celsius, 86 degrees Fahrenheit, Menthol or thymol, place crystalline menthol 50 g or thymol 15 g in a gauze bag on the top bars for 1 to 2 months. For better efficacy, place when the external temperature is around 21 degrees Celsius, 70 degrees Fahrenheit. 1.3 Non-insect pests. Till now we have studied about the major insects causing considerable harm to the honeybees. There are many non-insects which are also causing equal harm to the honeybees. We shall study about these non-insect enemies of the honeybees in this section 1.3.1 Birds. In Asia, bee-eater birds are the most important honeybee predator. Bee-eaters are bright-plumaged insect-eating of the Old World. They belong to the family Meropidae divided into 3 genera containing 24 species. Bee-eater birds feed exclusively on airborne insects such as bees and their allies, Hymenoptera, comprising 80% or more of their diet, and thus they are of great concern to the beekeepers. 1.3.1.1 King crow and black drongo, Dicrurus macrocircus, king crow, occasionally visit apiaries on cloudy days and prey upon bees. The black drongo is a small Asian drongo and is a common resident breeder in the tropical southern Asia. This species is found in open countries and usually perches and hunts close to the ground. It is an aerial predator of insects and prey upon grasshoppers, cicadas, wasps, dragonflies and honeybees. The nesting of the black drongo is done during April to August. 1.3.1. To green bee-eater. Small bee-eater, Merops orientalis, do much harm in apiaries. These birds are distributed in the Indian subcontinent except eastern Assam. It is a migratory bird which come in swarms and predate on honeybees during brood rearing season. Breeding is usually in sandy soils, making shallow tunnels to a length of 8 meters. Besides common perching sites, the bee-eaters sit on the beehives, waiting for foragers of a mellifera. A flock of 60 birds consume about 709 bees per day from an apiary of 40 colonies. This predator is usually active during 10 to 13 hours. This species keep watch on passing insects from a vantage point like a bush, fence, post, electric post, iron, telephone wire, and catch the worker bees on foraging. It feed on Acerana indica, a mellifera and a dosata. The swarm of these birds can be managed by scaring 1.4 bear and other mammals. You will be astonished to learn that besides insects and birds, there are many other bee lovers in the nature, including human being. The honey is considered as nectar of the nature. Honeybee hives have often been found attacked by different animals such as bear. These animals climb on the tree and squeeze the hives to eat the honey. The bears are by far the most honey-loving wild animal. They flat out and destroy hives in minutes. If you are intending to establish your apiary near forest, sufficient protection for honey from these animals is required. The apiary can be protected by providing electric fencing around the apiary. 1.5 Let us sum up. Like any crop plants, the beekeeping also suffers due to its enemies. The honey is liked by many insects and animals. The honeybee's enemy consists of insect and non-insect. Before establishing an apiary, one should have all the assessment about the threats and their possible management practices. These enemies of the honeybees can be managed by adopting different approaches. You may use any other method if it is available in your locality besides those which are mentioned above. Thank you for watching. We will see you in the next video with the next chapter.